Thanks for having me, and thanks for organizing this workshop. As you have mentioned, I'm a big believer in NWB. I think it's a much needed step forward in the field. So what I thought I would do today in this half hour is to first give you a little bit of a background of the kind of experiments we do, so that you have an idea of what the needs are for a data format for our experiments. And then I'll talk to you a little bit about what our internal data formats are presently, which hopefully will eventually be accommodated by NWB. And then at the end, I'll talk specifically in some detail about how we adopted NWB, which groups we used, which variables we used, and I'll also make some comments on some difficulties, not difficulties, but some lessons learned related to sort of clinical data, like there are some required fields that we can actually not provide and things like that. And then, yeah, if there's anything unclear, please just interrupt me and speak up. I may not see if you raise your hand, so please just interrupt me. So before I get started into the data, I just want to briefly mention why we do these experiments. So one of the things that fundamentally fascinates me and that I would like to understand at the circuit and single neuron level is how it is that we can have this distinct experience of mental time travel, meaning we can think about a salient event in our lives, such as when you think back to your graduation, let's say from college, where that was, how you felt, what the weather was like, what somebody said, etc. It really does feel like something doing that, and it's an open question of how that works and how it happens, and specifically how we instantiate this problem, how we investigate this problem, is through something that you can see on the slide. We show series of images, and you would see a particular image the first time, then it's novel, it's being encoded together with some other attributes. And sometimes later I'll show you this picture again, and I would ask you, have you seen this particular animal before? And when you saw it the first time, did something come to mind, or did you learn something else, or where did you see it? And so that's the processes that we are investigating. And the ways we investigate these questions is using invasive recordings in humans, which we can do in a variety of scenarios. And today I will focus on the microwire recordings shown on the upper left. And this is just to illustrate that the various things we do, which are all centered around trying to understand the human brain, of course, you could equally well frame this problem around NWB, because all of these things either generate data or consume data. So for the purpose of this presentation, I could equally well replace the human brain here with NWB, which will be the centerpiece around which feeds all these different aspects of the work that we do in my lab. So just to get started, the kind of data that we worked on storing into NWB is extracellular recordings of two kinds. So these are both from depth electrodes. And let me see if I can make a laser pointer here. There we go. So these are the depth electrodes. And you see here at this red arrow, you see these little microwires coming out. These are 40 micron diameter microwires, which are insulated all the way to the tip. So they're high impedance, and they yield a signal such as this. So from this, we can record LFP, as you can see, and spikes, single neurons here after sorting. And then the other type of recording that's done routinely and that we do is so-called macro contacts. These are big metal contacts which are low impedance, and which yield a signal that's variously called either IEG or ECOG. So that's a field potential signal. And then, of course, based on this, we'll do spike sorting. We isolate single neurons, and all the results of spike sorting, as well as various quantification of the process and parameters, is of course something that should also be stored in the NWB format. And so before I go into the NWB itself, I'm just gonna give you a brief overview of one recent experiment that we did, just so you get an idea of the kind of data needs that we have for our format and how we go about storing this data. So the scientific question that we wanted to address here is, how do we make decisions about memories? So let's say I show you a picture of a person and I ask you, is this, does this picture show your mom? How would you make that decision? Obviously, the answer to that question is not in the picture, because the answer is different depending on who I show this picture to. So it's a decision about an internal state, about a memory. And one way to conceptualize this decision is that there would be a representation of the actual memory, the evidence somewhere. And we have isolated such neurons, which we refer to them as MS cells. And there are two types, the novelty and familiarity type, in the hippocampus. And if you listen to these cells, you can predict with high accuracy whether a particular person is looking at something familiar or novel. So the question is, how is that signal translated or transferred or used to make a decision? So we were looking for choice neurons. So choice neurons, which are here illustrated in blue, one way to conceptualize or hypothesize about choice neurons would be that they would function like this, in that they would integrate a rectified difference between these two. And for various reasons that I won't go into, a good hypothesis or a reasonable hypothesis that such neurons, among other areas, would exist in the medial frontal cortex, based on an extensive body of literature. So the recordings I'm going to show you now, in which we converted into NWB, are testing this hypothesis by recording from these 4 areas. 2 areas in the medial temporal lobe, amygdala, hippocampus, and 2 areas in the pre-SMA and the DACC. And each dot here is the location of a microwire bundle across different patients. And this information here, of course, is also something that should end up in the NWB file. So let's first look at one slide for behavior, because we lots of the challenges we had in adopting NWB, surprisingly, is actually related to behavioral data, not to electrophysiology. So here, what we did is the patient is looking at a screen where a series of things are shown, and this is a task switching paradigm. So the idea is that the patient would be looking at a picture such as this one here and would give a yes or no answer. But the cognitive task that the patient would be doing based on this picture would differ. So this way we could create a situation where both the sensory input and the motor output is identical, but the cognitive configuration of the brain is different. So here, what we did here, either we asked, have you seen this image before? Or we asked categorization questions such as, does this image show a fruit? Yes or no. And we also asked them to reply in 2 different ways, either by pressing buttons or by making eye movements. That's why we could further disambiguate choices from motor responses. And you see here some example data. So here, while they were pressing buttons, we asked patients to fixate. As you can see here, this is eye tracking data. And if we ask them to answer with eye movements, there's a closed loop paradigm. They would look to the left or to the right, as you can see here. And yes, and I don't know if this comes through for you or not, but okay, there's something flashing up here. And then you can see here that the patients do learn. So the same images, some small subset of image will repeat in several of the memory blocks. And you do see they show single trial learning. This is the first time they see the images repeated. So they're pretty high, and then their performance increases even further. So that's the behavioral data that we would like to store in the NWB file. And then, of course, we would like to store single neuron data. And I give you a little bit of a brief impression of what that data looks like. So one type of cell that you'll find quite repeatedly in this kind of experiment is a category cell, as you can see here. For example, this cell response, it's a visual response. It's a response to the onset of the stimuli if the stimulus contains a human face, but not a monkey face. So it's a human face cell. And so that's one of the things that we would like to store, of course, meaning category information or just ground truth information. And just to give you a glimpse here, there one finds the interesting effects of the configuration of the brain, which shows that these category responses are much, much stronger throughout the brain. Even here, this is a saturation, but using other effects, one can still show this. When one does memory retrieval. So these categorical representations are much stronger when we're engaged in deliberate memory retrieval. However, the tuning doesn't change. It's really just a strength increase. So the geometry remains the same. This is this generalization analysis. When we train a decoder in one task and test it in the other, they fully generalize. So the geometry doesn't change. It's really just a strength change. And just to give you one glimpse into the frontal lobe, in the medial frontal cortex, we have identified choice cells. You see here 2 examples of these choice cells. For example, this cell number 2 here differentiates between yes and no responses, but only if they're about a memory and not if they're about a category. Vice versa here. This choice cell signals yes versus no, but only if it's a category decision. And these neurons truly are choice cells. They don't represent the evidence because they predict the choice better than the ground truth. And then lastly, another element of the data that one would like to store in the NWB file, of course, is information related to the LFP or the phase of the spikes. And this is fairly complex here, so I briefly want to show you this here. So for example, one analysis that we wanted to do, that we did, is to record the spiking activity and LFP activity simultaneously in one area here in the medial frontal cortex and LFP in another area here in the hippocampus. So this, using this analysis, we could do then interareal spike field coherence. So ask the question, okay, spiking activity in the medial frontal cortex, is that related to a particular phase of the LFP in the hippocampus? And if so, is that modulated by task, which we would take as preparation for solving a given task, because the analysis here is done before stimulus onset during baseline. So the screen is blank, but the patient already knows for the upcoming stimulus what task they're supposed to do. And interestingly, doing this analysis, we found that the activity of medial frontal cortex cells, and in particular of choice cells, change their phase locking of their activity to theta band activity in the MTL by instruction alone. So the only difference here is what we told them they were about to do. And you see here there's a quite a pronounced change in the baseline spike, interareal spike field coherence. So with that, I will now go to the more technical part. So how do we record and store all this data, and how do we put it into NWB eventually? So our setup is like this. So we this particular data that I talked about, it's recorded using an electrophysiology system, which in our case is Neuralink, and 2 other systems, which are both doing the acquisition. So there are 3 systems in total. There is a computer that runs the psychophysics in PsychToolbox in MATLAB, and there is the eye tracker. So the way that we synchronize things is everything is triggered by the psychophysics system. This psychophysics system sends TTL, sets them on, sets them off, set button presses, and so on, simultaneously the same to the Neuralink system over TTL through parallel port and over Ethernet through the iLink system. And that way we can synchronize the 3 different clocks of these systems, which are totally independent. And then the internal data formats that are presently used. So the psychophysics system stores behavior, meaning what was shown, which buttons were pressed, and such. And the eye tracker stores locally the position, the gaze position, which is also shared in real time here with the psychophysics system, but it's stored here. And then the Neuralink system stores the events, which are called EV, which are the TTLs, and it stores the continuous recordings. And what the Neuralink system outputs is then used as a source for spike sorting. And as a result of spike sorting, we have spike times, cluster IDs, waveforms, spike quality metrics. So that's all the data that ideally would all be incorporated into an NWB file. So the starting point that we have is this before exporting to NWB, is that the electrophysiology, everything is stored, the timestamps, they're all in microseconds, but with an arbitrary start point. So they are there, it's not an absolute time, and that doesn't in fact exist in that data. And everything is stored reference to this. This is the master clock, is the Neuralinks. All data is referenced to that. So the sorted neurons, so also the events are referenced to that same timestamp with TTL values, and we have conventions such as the ones listed here. Then the sorted neurons, after sorting, we have 1 file per channel, channel X, where there is a list of timestamps, again in microseconds, with cluster ID, and then also each cluster ID has various other metrics associated with it. Continuous data is also stored like this, same timestamp, but then amplitude. Behavior, the local system stores additional behavior, but that's now on the system of the psychophysics computer, so it's a different reference frame. But it also stores the same list of events, so this way we can equalize the 2 timestamps. And it stores stimulus information, the video or the stimuli that are shown, and also important information about what experiment was actually done, which variant, and so on. And then, of course, we have subject information, demographics, various clinical information, mapping of channel numbers to recording electrodes, and imaging, if that could also potentially be exported, although we didn't actually do that. So now, for the last part, what I was going to do is going to briefly take you through how we took all that data that I just talked about and how we utilized NWB to store all that into a single file, 1 file per session. So I'll mention how we ended up doing this, and I'll have some decisions that we made along the way. And this is all documented in this data descriptor that I'm showing you here on this slide. So this is how these are the aspects of NWB that we ended up using for our data, and I'll show you a little bit of which fields and which groups we ended up using. So I'm going to go from right to left. So we'll talk about the units, how we stored the units, how we stored the stimulus, and the trials, and so on. So starting from the right here with the units, The way we ended up utilizing the NWB format is as following, and this of course is mostly standard, although there's some modifications with custom columns. But so just to go through this, we in the units table, we added all the units here, as you can see, and this is the standard how we reference to spike times. So the only change we did here is we convert to seconds instead of microseconds. And as I'm sure you've already seen, internally, all spike times are stored in a single column, and the different units are just indices, ranges into this large table. And then further in the unit table, we added here a variety of custom columns, which the unit table allows. As you can see, we wanted to still preserve what the original cluster ID was if we needed to go back into our spike sorting, and some quality metrics that we wanted to preserve so that one can sub-select units based on certain criteria. And another thing we did here, we stored the mean waveform of that cluster during two different parts of the experiment. And then here we use this reference into the electrode table to show, for example, here is two units isolated on the same electrode number 0, which then indexes into this group. And here, what we did is the XYZ, we used actually MNI coordinates. So that comes from our MRI localization. So that's where dots were plotted. We used also the area label here. We stored what the original hardware channel was, because that's sometimes useful information that we need. The impedance of these electrodes, we don't know individually, but then we also separated them into micros, so those are the microwires high impedance, and macros, those are where there's just LFP. And let's see, lastly here, then we used the electrode group to identify what the actual electrode is that we used, the recording system, and the full text of the area to map the abbreviation to the area. So that's how we stored the units. And then the stimulus information. I'm just going to show you some abbreviated MATLAB code of how we, like MATLAB-ish pseudocode, I should say, how we exported the various stimulus information. So in this experiment, what's shown is a sequence of images, which are colored. So we used this particular way, we used this optical series, which probably wasn't exactly meant to store stimulus information, but this ended up working well. Maybe Ben can make comments on this later. So this is how we stored the stimulus information, and also various other things about how large was it on the screen, what's the distance of the subject to the screen, what's the dimension shown on the screen, all kinds of information that otherwise would never be explicitly encoded. So this was very helpful. Moving further along, the intervals group, which stores trial data, the responses, and the ground truth in our case. So this we use this time interval structure, which has a start time and then various sort of custom columns that we added to identify things such as what's the category of the stimulus, like in that case there would be monkey face, human face, fruit, and so on. What's the ground truth? Was it new or old? And what's the response given by the subject, which here is a 1 to 6 response. So this is how we stored the ground truth and the actual behavior of the trials and when they were shown. And then lastly, the more raw data, the acquisition stream on the left. We used in this particular thing, we used 2 acquisition streams. One is the events. So this maps time steps with TTLs. And then we have another acquisition stream, which we call experiment IDs, because in one given session, there may be multiple experiments, such as a learning phase, a recognition phase, or another recognition phase. We call that experiment ID. So that way we could map subsets of events into different experiments. And of course, not shown here, but also in the acquisition group, we could also add the continuous LFP using the elliptical series group. And lastly, I was going to mention this here. So we have some internal conventions of how to map which TTLs mean what and which experiment IDs mean what. And we use the description field for that. So these tables here are in the data descriptor, but they're also in the description field. So it should be fully self-contained. And then, so lastly, just let me make a few comments of things that were important to me or that I learned while doing this. So one of the things that is really important to me is that we don't bind ourselves to a specific programming language or operating system. So NWB was really ideal for this because some of us use Windows, some people use Mac, some use MATLAB, some use Python. So in using NWB, I really wanted to make sure that we don't abolish this key feature of our code base, because that really would limit things. Then HIPAA compliance, I mentioned this, we did skip some fields which are actually technically required. Because, for example, one piece of information that one is not allowed to reveal is the date of somebody's hospital admission. That's protected. So we'll put that. One thing that we found really, really helpful is round-trip testing between Python and MATLAB. It actually revealed many little quirks of things that weren't quite done correctly, that if you stay in the same programming language, you would never notice. But if you try to read it in another programming language, you're like, wait, this is actually ambiguous. So we found this to be very helpful. And for that reason, in that particular data descriptor, everything is done in both languages. And to really prove that this is the case, export in one, read in the other, and vice versa. In my mind, that's really the proof that a data standard works. But if you do it only in one programming language, not really, right? Yeah, exporting to NWB, I'm sure many of you have experienced this, was actually was really a good forcing function for us, because it forced us to do various things that otherwise it would never clearly define. And yeah, but lastly, one thing that we did in many instances is to actually skip exporting the stimuli because the files are too large. So if you wanted to do like high performance computing and farming out to a cluster, the files would just be too large. So we focus them on, basically we have two versions of the files, one with and without stimuli. So with this, we'll conclude. Here are again some of the links. You can download this data from Dandy. And if you want to play with the exporting, the actual raw data is also provided on OSF, as is the code on GitHub and the actual data descriptor. So thank you.
All right, thank you so much, Ueli. I think maybe we have time for one question. You came in just on the mark, so I think we might have time for one if people have any questions.
Sure, yeah, please go ahead.
I see John has his hand raised. Go ahead.
Yeah, hi, thanks for the talk. I'm just curious, so a lot of the things you showed about how to set up a pipeline and how to connect all the different parts, these are things that most labs already have, right? And sort of the second to last point you had at the end where you said what you're basically doing is you have your pipeline, as far as I understood, and then at the end you're basically just exporting everything into NWB. So how, I guess I have two questions about that. I don't know if we have time, but one is sort of how much time has it taken you? So this whole process, you know, to get to the point where you can export everything into NWB in a nice way? And the second one is, is there any reason why you would not basically replace or go with NWB from the start? So, you know, is this just a time issue, or is there another reason for that?
Yeah, thank you for those questions, Sean. Yeah, so that's definitely where we want to end up in the future. But so we decided to do this as a first step because it allows to do a kind of a smooth adaptation without having us to change the like spike sorting and all those things. So because right now our spike sorting tools they just don't take NWB as input and output. So, but that's definitely where we would like to be eventually, to make things smoother, because this is one of the common things you have. Somebody new comes in the lab and you want to give them some data, and then they have to first discover your data format. So that's one problem that eventually I would really like to solve with NWB, so that I can say, okay, here's NWB file, and that's how you can read things. So to answer your first question, that the time investment was actually, I'm sure many of you had this experience, it's actually quite substantial, but for much of which has nothing to do with NWB. It's just, there's this, it really forces you to, it's like, wait, actually, what was the distance to the screen? And, you know, there's just so many things which are basically implicitly assumed, but which we never actually formalized. But it was actually very helpful also for me, because one of the things if you have a lab, you have so much flux of people. Then if somebody generated a data set that left 3 years ago, you know, I think it's really good to have that forcing function. That's why I put it now at the end of the project, because then I will, at least at the very end, I force everybody to do this.
Right.
Or myself, I should say.
Yeah, well said, Ueli. I would also like to add that Ueli was such an early adopter that many of the tools that we have now, that we're teaching you now, were not in the state that they are now when they started this project, and certainly not when they started the collecting the data and analyzing it for the eventual publication. So these days, it might be a little bit more feasible to take a project and bring it into NWB earlier on. For sure, yeah, and that's definitely what we are going to push towards. Yeah. Great. Well, thank you so much. Thank you for having me. Wonderful talk. I think that really was a nice overview of how to convert that kind of data. And I hope it's really helpful to the people who are listening now. And we'll also, if you don't mind, be putting it up on YouTube so other people can benefit from it as well. Yeah, of course. And the actual the export script that we used is on GitHub, so you can use that as a starting point if you want to export your own data. I'll put the links to those in the YouTube description, so people can just get rolling right away with that. Great. All right, great. Thank you so much, Ueli. Thank you, Ben.